So our final speaker tonight is Associate Professor Michael Nissen. So we're very excited to have Michael back in Brisbane after a time away. So Michael's had over 30 years of experience in clinical medicine and ID and paediatrics and clinical laboratory microbiology. And he's got a particular interest in the epidemiology of vaccine-preventable diseases. Michael is our first ever director, our inaugural director of research at the Prince Charles Hospital. So it's fantastic to have his leadership and experience to guide us. And prior to taking up this recent role, he was a principal medical officer at the TGA in Canberra and been director of the pharmacovigilance of COVID-19 vaccines there. And he's also worked in industry overseas, as he may touch upon. So thanks, Michael.
But thanks, Andrew, and the CRE and Heidi for asking me to chat. It's actually nice to see you all again, old friends and colleagues. And also, I'm really quite glad actually not to be talking about COVID for a change. So what I wanted to cover tonight was just some recent successes in respiratory pathogen development and touch upon where previously they have failed, but also now those failures have been turned into success. I have a conflict of interest in that I worked for GSK for 6 years, and one of the vaccines I'll be talking about tonight is actually a GSK product. So I don't need to tell you about the importance of acute respiratory tract infections, including COPD and asthma, among the most common causes of mortality in the world. The burden is mainly in children, and the etiologies I'm sure you're all familiar with in terms of RSV, rhino, influenza, adeno, and metapneumovirus. When we talk about bacteria, we're mainly talking about Streptococcus pneumoniae, globally Hib, and occasionally Staph. If we're thinking of other community-acquired pneumonias or respiratory tract infections at a global level, we can't forget about MTB and also non-typable H. flu and Moraxella, which are the prime etiologic agents in COPD exacerbations. So vaccines, other than clean water, are the most effective public health measures that we have. And a recent review by Julia Gutenberg and Brian Haynes in the New England Journal actually just looked at what the innovations had been in vaccines and licensure by the FDA since 1970. Oh, sorry. That was supposed to be, went ahead. Apologies about that. And I've highlighted here the ones that are respiratory pathogens or vaccines. So you can see way back from 1977, where it was the actual first polysaccharide pneumococcal vaccine, right up until the late last decade. There's been numerous developments in respiratory pathogen vaccines, and some of them have built upon previous knowledge, but also previous failures. In particular, when we think about the polysaccharide vaccines for pneumococcus, wholesale vaccines for pertussis, though some people would argue that that actually was a success compared to acellular vaccines at the global level, and also influenza vaccines. So I've listed here what I think are some of the successes of respiratory pathogen vaccines. You may debate the merits of some of these, but certainly when these vaccines are used in epidemiologic, public health and large-scale vaccinating programs, they are effective against respiratory disease. And what I wanted to touch upon tonight was actually two pathogens, RSV and TB, that have had historic failures, but now hopefully these have been turned into future successes. So the first paper I want to talk about was published in April this year in New England Journal. And this was actually the use of a RSV vaccine in pregnancy. As you know, vaccine studies in pregnancy are very complicated, one in terms of recruitment, but also potentially standardizing recruitment across countries, but then also monitoring for both maternal adverse events, but also follow-up of the fetus and the infant, particularly for RSV disease in the first 2 years of life. So one of the main barriers about RSV, and I'm sure Chris touched upon this in terms of RSV vaccine development, was stagnated for basically the last 30 or so years following the deaths of infants that were given whole virus, formalin inactivated RSV virus. This then actually led to antibody-dependent enhanced infection, and unfortunately, there were infant deaths. And the main reason for that is this is the post-fusion form of the F protein, which is involved in attachment. And it's this antibody that was potentiated with the inactivated formalin vaccine because it was in its post-fusion state. Due to some very elegant work and molecular confirmation work by Barney Graham and his group at the NIH, they were actually able to understand that the pre-fusion version of the F protein was the one that actually had the excellent epitopes for neutralizing antibody that prevented RSV virus entering or attaching and entering into respiratory epithelium. The difference between the pre and the post-fusion is basically energy. So this is really like a safety pin, and it's in a high energy state, and it wants to move from this state to this state. And in that process, these really highly antigenic epitopes become buried or hidden inside the head of this molecule. So they were then able to, through site-directed mitogenesis, stabilize this pre-fusion protein so it prevented from going, into this state. Now, you may be familiar with the work of Paul Young here at UQ and the molecular clamp. And the molecular clamp is very similar, and Paul has used the molecular clamp to stabilize RSV pre-F. As you know, there are certain problems with that in terms of the series or the clamp that is potentially used, but it's exactly the same, I suppose, approach into maintaining this pre-fusion shape. So this was an overview of the study. So it was a phase 2B. It was conducted in the US, Chile, Argentina, and South Africa. And it was given to women in 2 doses. And it was mainly there for safety and antigen selection. It really wasn't an efficacy study at all. And I'll go through the results individually. And then the infants were followed up following delivery. And the vaccine was given somewhere between 24 to 36 weeks gestation. And certainly it's been shown that this is the prime time in terms of immunoglobulin transfer across the placenta to protect the infant. So this was an overview of the study. It was 406 women that were randomized and into the 2 treatment doses. And over that, you can see here how many babies were delivered in each of the treatment groups and then the placebo. So again, you can just see the complexity in this study in terms of the detailed follow-up that's required to, one, assess safety, but also potential antibody titers. This is just a panel that really looks at injection site reactions in the mother between the different groups, and then systemic reactions. And as you can see, that there was quite a degree of similarity between the different dosages, though it was more marked than placebo for certain things such as redness and swelling, and the systemic effects tended to be a little higher compared with placebo. The other main concern is fever in this group, and there seemed to be fever, surprisingly, with the lower antigen dose. And the antigen of interest was delivered with or without an adjuvant in terms of aluminium hydroxide. When we look at the neutralizing titers to both RSV A and B, and this is an important aspect of RSV studies, because serologically, RSV is recognized into 2 distinct types, A and B. And then within A and B, there are 2 genotypes, A1 and A2, B1, B2, etc. But A and B is what's important in terms of an immune response. And you can see here that compared to placebo, there was a higher rate of neutralizing antibodies, and not a lot of difference between the different dosages. Apologies, this is a sort of a busy slide, and though this study wasn't actually powered for efficacy, what we have here is detailed adverse events in the maternal immunization, and you can see that actually adverse events either of a severe nature in both the maternal participants or in the follow-up of that outcomes were very similar. This is actually quite relevant because there was another RSV candidate vaccine that was at a similar stage of development and has been paused because of poor infant outcome in terms of low birth weight. And that's potentially now is a topic of further analysis, both within a subset of these infants, but also that other study. As I said, this wasn't powered for efficacy, but what it actually shows in the follow-up of these infants, that even though we're talking very low numbers here with extremely wide confidence intervals, it did seem to be effective, but this really wouldn't be classed as significant, and really requires now the phase 3 trial that has commenced. RSV also causes a problem in adults, and those particularly older than 60, but especially those who have chronic heart or lung disease. And certainly, unfortunately, from the age of 50 years on, our T cell function begins to decline, and we're all potentially at risk at T cell mediated or dependent infections, and herpes zoster and shingles is the prime example, but it applies to respiratory tract infections as well. So the RSV older adult vaccine is exactly the same as what's used in the maternal cohorts or study that I just previously described to you, but it actually has an adjuvant added to it. And this is a specific adjuvant which has both monophospholipid and also saponin. And it's the same adjuvant that's actually been used in the RTSS malaria vaccine, and but is different from the adjuvant that is currently used in the herpes zoster vaccine, that is the killed vaccine. So this is just actually results, and these are unpublished, but these results were presented last week to the ACIP in the US. So that's the Vaccine Monitoring or Technical Advisory Group for the CDC. And the results are actually quite impressive in terms of protection against overall RSV disease of 83%. And also, more importantly, with severe RSV disease of 94%. Similar against both subtypes of RSA and B, most effective in those that are 60 to 79 years of age, which is no surprise. The 80-year-olds really is not significant, and we're only talking about 2 versus 3 cases in that group. Comorbidities were also covered in this in terms of low and high risk disease, and also greater than 1 comorbidity, so both either heart and lung together, and then assessed on frailty as well. So this is a very promising candidate, and the aim is that this will probably be going to FDA registration by the end of this year, and will hopefully be available next year. Of course, I think the most interesting aspect of this study is going to be really in immunocompromised individuals, and those particularly transplant patients, and also other patients, hematology, oncology patients who are immunosuppressed. So the last vaccine I wanted to talk to you about was actually a therapeutic TB vaccine. And this is the final analysis of a trial that had been conducted from 2010 to 2015 in Africa. So countries included South Africa, Kenya, and Uganda. And it's been the subject of 2 New England Journal papers. This is the last one, 2019, but there was also preliminary results in 2018. So this study is really looking at the 3-year endpoint, while the previous one looked at 2-year follow-up following this vaccine. So in an interim analysis, the efficacy in the phase 2b study was 54%, and overall in this study, and that was at 2 years, and it's 3 years, it had fallen slightly to 49%. But this reaches the approved level of the WHO in terms of a potential vaccine that could be used in a widespread population vaccination campaign. So what is the, So what is the vaccine? It's actually a fusion protein of MTB32 and MTB39. And the 32 protein and spans, or is it caps the ends of the MTB39. And this is thought to be the prime proteins that are involved in T cell stimulation. Again, this is with an adjuvant, the ASO1 adjuvant, so it's monophospholipid and saponin. And again, this adjuvant has particular, I suppose, targeting towards T cell responses. And so what's important in this paper is the primary endpoint was actually the prevention or the development of definite pulmonary TB disease that wasn't associated with HIV infection. So this was actually giving the vaccine to those individuals in high endemicity countries aged between 18 to 50 years of age who were quantiferon positive, though sputum negative or PCR negative. So they were at high risk of eventually developing TB disease. There were secondary endpoints or second definitions, which I've listed here, and certainly the full analysis goes into the efficacy in this, but this, as I said, was the main endpoint in HIV negative patients at high risk for getting disease. So this is really just the Kaplan-Meier plot, and what you can see here is over the 3 years, the probability of freedom from tuberculosis disease was, as I said before, ended up around 50% over that time period, while those who received placebo, it was in the mid-20s. So there was a statistical significant difference between those two groups. If we look at the number of cases, according to their age of enrollment, you can see most of these individuals were young adults in their third decade of life, though there were some that were more elderly, and then there were some that were between 18 to 20. Again, a busy panel, but really just showing you the antibody titers, the GMCs, against this fusion protein compared to placebo. And also this was maintained over the 3 years of the study with very little decline, particularly in the last 2 years. This panel is looking at positive CD4 positive T cells, again, the vaccine versus placebo. So you can see that CD4 T cells are being stimulated. The expression of specific immune markers in terms of interferon, tumor necrosis factor, IL-2, and CD40L, again, showing a high degree of persistence over this time period. No major differences between the countries, and the final analysis was only done between South Africa and Kenya because that's where the blood was collected for these specific markers, and then just purely looking at interferon gamma between these two countries as well. So not a lot of difference between the different countries. So if we look at, again, the table, and again, this is looking at all the definitions, but if you just concentrate on the first definition, which is those who are at risk of TB disease, quantiferon positive, and protection from developing pulmonary disease, you've got a vaccine efficacy of close to 50%, but fairly broad range of confidence intervals from 12 up to 71. But again, as far as the TB vaccine goes in this high-risk population, this is a really impressive, encouraging result. Don't worry too much about this. With the IFA, this is just looking at the demographics of the recruits in terms of comorbidity, sex, country, age, and there are not a lot of major differences between the two. I've mainly pointed out to you the age differences, and some of these individuals had already been given BCG, but they weren't thought to influence the final results. So this paper actually was judged the paper of the year for 2019 by the New England Journal, mainly because of its potential at a global level to prevent the prevention of TB and hopefully then NDR. What I think is even more impressive is even though this was an industry-sponsored trial, they subsequently then passed over the patent to this vaccine to the Bill and Melinda Gates Foundation. And they are then going to now use this in a phase 4 trial, similar to what's been done for the malaria vaccine in sub-Saharan Africa. So hopefully that's given you some encouragement for the future. I think we will have an RSV vaccine for older adults within the next 12 months. Very likely that we will have a TB vaccine within the next 3 to 5 years for use in our population, and hopefully in the Western Pacific and Asian region sooner than that. And maternal RSV is probably about another 2 to 3 years away. So thanks.
Thanks, Michael. That's great. I might just get to stay close. People could probably hear you from the seat, actually, Michael. So I'll just see if there's any questions. Claire North.
Is an efficacy of giving vitamin D treatment to person with TB.
You mean?
So just itemized with right hand, you know,
That
random TB treatment to eradicate.
Yeah, no, good question, Claire. I think that wasn't done, of course, as part of the study. That will be probably one of the things that WHO and planning wishes to do. As you know, though, the distribution of those short course regimens are complicated in terms of compliance, but also accessibility. So this is a 2-dose vaccine that can be given as part of other mass campaign vaccines.
What's the interval?
It's from memory, I think it is 1 month.
It's just now they've got that 3 HP, just once a week for 3 months. The prophylaxis stage is one.
Yeah. Michael, just for interest, the WHO cut off, you said for a useful vaccine, I think you said 50% met the criteria. What's the cutoff they use, Is it 50%?
I think that was sort of an expert panel's opinion in terms of based on what would make a difference in a way in the Pacific country. So it's really a multifactorial.
It's very exciting, I guess, and not trying to put cold water on anyone. I'm just about practicalities, as I'm sure others are. Obviously, the study enrollment, they had a positive quantifier on gold. So rolling it out to Timor-Leste, where people work, where people may not have that, is going to get a dilution of the efficacy at a population level. But Claire, how many people in Timor would have a positive quantifier on gold or mean to? Most populations. So you wouldn't need it to discriminate, really. Be happy to, And the other thing was about HIV, they were excluded from that study, Michael? Yes. Okay. So that was a fantastic talk. I'm just looking at questions from people online. So you're very upbeat, Michael. It's nice to finish on a positive note, which is good. That's what it should be. Because it's a lot to celebrate from all our speakers. So